All right, today we're going to dive deep into a really fascinating molecular mystery. Inside every single one of your cells, there's a tiny machine working with unbelievable precision to copy your DNA. But with so much on the line, how does it avoid making a single critical mistake? Well, let's find out.
Yeah, it's a really good question, right? DNA polymerase has literally one job: build DNA. The problem is, it's surrounded by the building blocks for DNA and its very close cousin, RNA. So how in the world does it pick only the right ones every single time? That is the puzzle we are solving today.
Okay, so to get to the bottom of this, we've got to meet the key players first. And you can think of them like twins, nearly identical, except for one tiny, tiny little detail that, well, it changes everything.
So on one hand, you've got DNTPs. These are the right guys for the job, the correct building blocks for DNA. And on the other hand, you've got RNTPs, which are used to build RNA. And they look almost exactly the same. So the question is, how does our master builder, the polymerase, tell them apart with such perfect accuracy?
Well, the whole secret, the entire key to this puzzle, it all comes down to one specific spot on their sugar backbone. It's a place called the 2' carbon. And believe me, this tiny spot is where all the magic happens.
Okay, so let's zoom in on the DNTP, our DNA building block. If you look at that 2' carbon spot, all it has is a simple, really small hydrogen atom. Just a single H. Keep that in your mind, it's super important.
Now, let's look at its twin, the RNTP. At that exact same spot, the 2' carbon, it doesn't have just a hydrogen. Nope, it has a bulkier hydroxyl group. That's an oxygen and a hydrogen bonded together. You know, an OH group. Now, that might not sound like a big difference to us, but trust me, at the molecular level, it is a huge deal.
All right, so we've got our setup. 2 molecules. One's just a little bit bulkier than the other. So now, let's bring in the hero of our story. Let's unveil this absolutely brilliant and elegant solution that DNA polymerase uses to tell them apart.
Okay, so deep inside the DNA polymerase, in a place called the active site, this is where all the chemistry happens, there's this amazing feature called a steric gate. Honestly, the best way to think about it is like a bouncer at an exclusive club. It's super picky. It's only going to let in the molecules that have the perfect shape and size. So what is this gate even made of? Well, it's actually formed by some of the enzyme's own amino acids, specifically really bulky ones like tyrosine or phenylalanine. They're part of the polymerase, and they just fold themselves into this perfect shape to create a very, very specific size doorway. Okay, the stage is set. Let's see this brilliant mechanism in action. We're going to watch as both of our twin molecules try to get past this molecular bouncer. First up to the velvet rope is the RNTP. Remember, this is the RNA building block. It walks up to the active site, trying to get into the DNA building party.
And denied! It can't get in. Why? Because that bulky hydroxyl group we talked about, that OH group on its 2' carbon, it literally bumps right into the bulky amino acids making up the gate. It's a physical collision, a steric clash. And because of that clash, it just doesn't fit. It's physically impossible for it to get in there correctly. The gatekeeper did its job, and the RNTP gets rejected, sent packing.
Okay, round 2. Now it's the DNTP's turn. This is the one we actually want, the correct building block for DNA. It walks up to that very same gate, and look at that, it slips right in. No problem at all. Since it only has that tiny little hydrogen atom at the 2' position, there's nothing bulky to bump into the gate. It fits perfectly, like a key sliding into a lock.
So the DNTP gets the green light. It's in. It gets added to the growing DNA strand, and replication just keeps on chugging along, smooth as can be. It's all because of that perfect, elegant fit.
So let's just break it down one more time. The RNTP, with that bulky OH group, it clashes with the gate. It's rejected. The DNTP, with just that tiny hydrogen atom, it slides right through. Accepted. It's that simple. Just a brilliant, purely physical filter that is ridiculously effective.
So there you have it. Puzzle solved. But let's just zoom out for a second and really appreciate why this little molecular trick is so unbelievably important for all of life as we know it.
Here's the bottom line. This steric gate mechanism, this is how DNA polymerase protects the integrity of our genetic code. It's not magic, it's physics. By physically blocking the wrong parts, it makes sure that our DNA stays DNA, and it prevents errors that could be absolutely catastrophic for our genome.
And just
think about this for a second. This whole selection process, this bouncer at the molecular club, it's happening inside you, right now.